Namaskaram. My name is S. Ashok Kumar. Welcome to my channel. Now, this part, I am going to continue the structure of proteins. In the last video, I explained about the primary and secondary structure. Now, this video, I am going to explain tertiary structure. Actually, tertiary structure is the completely folded three-dimensional structure of polypeptide chain. Stabilized by disulfide bonds. So what I said, tertiary structure is the completely folded three-dimensional structure of polypeptide chain. What I said, secondary structure. Secondary structure is the intermediate folding. Tertiary structure is a completely folded three-dimensional structure of polypeptide chain stabilized by the disulfide bonds. Here, this point you should remember. Which amino acid, which only amino acid responsible for the formation of disulfide bond? Immediately you have to tell cysteine. Cysteine is the only amino acid responsible for the formation of disulfide bonds in proteins that I will explain. Wait. For example, here I wrote completely folded. Now, see this one. This is a completely folded. This is a consider N terminal. This is a consider as a C terminal. This completely folded. For example, here it is a disulfide bond. Okay. So this is a completely folded three-dimensional structure, that is we are calling as a tertiary structure of polypeptide chain. Here now next point I am going to explain about the disulfide bond. For example, if you take this is a one polypeptide chain. Consider this is a CH2SH, one amino acid I am showing here. This is a consider polypeptide chain 1. Polypeptide chain 1. This is a which amino acid? Immediately you have to understand the R group. R group is a CH2SH. Means this amino acid is a cysteine. For example, you take one more polypeptide chain, one more cysteine I am showing it here, CH2SH. This is a second polypeptide chain. Here disulfide bond is formed by very simple. This is a SH. Here H, both are removing. See this one. There is a bond formed between the sulfur. This bond formed between the sulfur, this one is called as a disulfide bonds. Okay na? Here I showed disulfide is bond formed between the two polypeptide chain. Now you can call this is a interdisulfide bond. If the same disulfide bond formed within one polypeptide chain, that is called as a intradisulfide bond. The next one, here when I showed one amino acid I called as a cysteine, this amino acid I called as a cysteine. Now, after formation of the disulfide bond, if you observe, this is together called cysteine, C-Y-S-T-I-N-E, cysteine. Cysteine, what is the spelling? C-Y-S-T-E-I-N-E. That is the individual amino acid. Once a disulfide bond is formed, two cysteine together is called as a cysteine. Okay, this point you understood. The next very important point for the tertiary structure is a natural or functional form of simple proteins. This already mentioned in the concept. Tertiary structure is the functional form of protein or natural structure of the protein. Here what I mentioned, tertiary structure is a natural or functional form of the simple protein. What is the meaning of simple proteins? You know, if a protein made up of only one polypeptide chain, those proteins are called as a simple protein. Proteins made up of only one polypeptide chain, if it is there, those proteins are called as a simple protein. So simple protein, what is the functional form? That is a tertiary structure. Tertiary structure is the functional form of simple protein. So many examples are there. For example, if I called myoglobin, this I will explain. One more example you need to remember here for our syllabus, that is active site of enzymes. This is there in the NCERT, so important. Active site of enzyme due to tertiary structure of the protein. This point important. Active site of the enzyme due to tertiary structure. Okay, this is about the tertiary structure. Now comes to the quaternary structure. Quaternary structure. What is the quaternary structure, you know? Quaternary structure is the functional form of multimeric proteins. Multimeric proteins. Here I will give the good clarity to you. Here I said simple proteins. Here I am telling the multimeric protein. What I said simple protein means a protein made up of only one polypeptide chain. Here I used multimeric protein if a protein is made up of more than one polypeptide chain. Those proteins are called as a multimeric protein. For simple protein, what is the functional form? Tertiary structure. For multimeric protein, what is the functional form? Quaternary structure. Okay, na? For example, in our syllabus, only one very important example, frequently asking example, that is a hemoglobin. I will write A. Hemoglobin in adult. To discriminate that one, HbF. Hemoglobin in fetus. When somebody called hemoglobin, definitely that belongs to Hba only. For example, Hba, if I want to write, that is alpha 2 beta 2. HbF, that is alpha 2 gamma 2. Take this one as a standard Hba. Here I wrote hemoglobin in adult alpha 2 beta 2. What it means is, in this hemoglobin there are 4 polypeptide chains. 2 alpha polypeptide chains, 2 beta polypeptide. So how many total polypeptide chains? 4 polypeptide chains. Means more than one polypeptide chain. If more than one polypeptide chain is there, that protein is called as a multimeric protein. Now I am asking, what is the functional form of hemoglobin? Is it a tertiary structure or quaternary structure? That is a quaternary structure. One more example I will tell you. For example, alpha 2 beta, beta prime sigma. This one belongs to, the name of this protein is RNA polymerase. As we know, this enzyme mediates the transcription. Here how many polypeptide chains are present? Alpha 2. Beta, third poly, 4 and 5, sigma. Here there are RNA polymerase structure concept, 5 polypeptide chain. That means RNA polymerase belongs to multimeric protein. Then what is the functional form of RNA polymerase? Is it a tertiary or quaternary? Definitely quaternary protein. That is why, what you need to remember, for simple protein, the functional form is a tertiary. For multimeric protein, the functional form is a quaternary. That is why, if I ask, what is the functional form of proteins, if I ask, without confusion you have to tell, sir, that is a tertiary or quaternary. Tertiary structure is a functional form for the simple protein. Quaternary structure is a functional form for the multimeric protein. In multimeric protein, in the quaternary structure, you should remember only hemoglobin that is there in our syllabus. Now I will explain about the hemoglobin. What I said, hemoglobin consists of 4 polypeptide chain. Yes or no? Now I am asking you, how many heme molecules associated with the one hemoglobin molecule? Each polypeptide chain associated with the one heme. So total how many polypeptide? 4 polypeptide chains. So how many hemes are there? 4 hemes. How many ions are there? Each heme consists of 1 ion. So how many hemes? 4 ions. So 4 hemes means 4 ion. Which oxidation state? +2 only. Then each ion associated with 1 molecular oxygen. So how many molecular oxygen total binded by 1 hemoglobin molecule? That is a 4. That is why one hemoglobin molecule consists of 4 polypeptide chains, 4 heme groups, 4 ion, and maximum associated with the 4 molecular oxygen. That is a structure. This is a detailed structure of primary, secondary, tertiary, and quaternary. Different levels of the protein structure. Now I am telling the same concept. Different bonds responsible for the stabilization of the protein structure. One by one I will tell you. The first bond is peptide bond. Yes or no? Peptide bond responsible for which level of protein structure? That is a primary structure. The next one, hydrogen bonds, responsible for the stabilization of the secondary structure of protein. I explained very clearly. Third one is a disulfide bonds. These disulfide bonds responsible for the stabilization of the tertiary structure of the protein. Fourth and most important bond, that is hydrophobic interactions. Hydrophobic bonds or interactions. These hydrophobic interactions play vital role in the stabilization of the protein structure in the aqueous environment, because cytoplasm is nothing but aqueous environment. What it means, as we know, tertiary structure is the functional form. Yes or no? When you consider that one, tertiary structure, when it is a completely folded, when it is completely folded, the amino acid, you can see certain group of amino acid present on the surface. Remaining group of amino acid present inside of the protein. Here, the protein, the amino acid present on the surface, they are hydrophilic amino acids. Because these hydrophilic amino acid side chains, why I am calling side chain? Because carboxylic group and amino group, they already involved in the peptide bond. Only free one is a R chain. So the R groups of the hydrophilic amino acids associated with the surrounding water. Yes or no? The inside amino acids that are hydrophobic. At any cost, these amino acids cannot associated with the water, but they are present within the aqueous environment. That is why the R groups of hydrophobic amino acids, they are present inside and they interact among themselves in order to increase the stability of the molecule. Those interactions present among the hydrophobic side chains of the amino acid in the core region of the protein, they are called as a hydrophobic interaction. These 4 are the bonds responsible for the stabilization of the different structures of the different protein structure. The next concept after this protein structure, now last with respect to the proteins, I am going to explain about denaturation of proteins. Here the name directly indicates what is the denaturation of proteins. D means what? Removing. What is the natural structure of the protein? The natural structure of the protein, you have to take tertiary structure. Because tertiary and quaternary structure both are functional form, both are natural structure. But from our syllabus, what is the only example for the quaternary structure? Only hemoglobin. Remaining all proteins you have to take as the functional form is a tertiary. That is why I am telling denaturation of the protein means removing of the natural structure of the protein, or you can call it removing of tertiary structure of protein. I will explain. Removing of tertiary structure of protein. For example, or normal or natural form of protein you can extract, then you keep in the test tube. After keeping, start heating. Here the heat is the denaturating agent. So now consider this is the tertiary structure of the protein. Okay. When you started heating, disulfide bonds undergo breakdown. Then tertiary structure converted into secondary structure. Then these hydrogen bonds also will break. then convert into primary structure. Means this is amino acid 1, amino acid 2 linked by peptide bond, amino acid 3 linked by peptide bond. So what is happening during the denaturation, if you ask me, in general I can tell conversion of tertiary structure of protein to the primary structure a denaturating agent. Okay. In another way, if I want to define, during the denaturation, what happening? Disulfide bonds undergo breaking down. Yes or no? Hydrogen bonds breaking down. But peptide bond remains the same. That is why peptide bond belongs to covalent bond. This disulfide bond and hydrogen bonds belongs to non-covalent bond. That is why what you need to remember as a point of concept, what is happening during the denaturation means all non-covalent interactions associated with a molecule undergo breakdown. Only covalent bond remains the same. With reference of the protein, what is a covalent bond? That is a peptide bond. Here, during denaturation, what I said, protein losing the tertiary structure of protein. Losing the tertiary structure means losing biological function. Why? Because what I said, tertiary structure is the functional form. That is why losing the tertiary structure of the protein, indirectly that particular protein losing the biological function. For example, what you need to remember the example, at a high temperature, remember more than 60 degrees centigrade, at a high temperature, enzymes loss function by denaturation. This example you should remember because enzymes presented in the same chapter. At a high temperature, enzymes lost their function by denaturation. This is the concept of denaturation of proteins. Till now I explained about the molecule, about proteins. Next video I am going to start about the enzymes. Till now, if you understand the concept of proteins, you can like and share to your friends. You can subscribe our channel. If you have any doubts, comment below. Definitely I will respond to your questions. Thank you.